Hello everyone, and welcome to the seventh edition of the Brazilian Student Consent Symposium. My name is Matheus Veiga, and I'm part of the organization team, and I'm honored to be here with you. This year, our theme is addressing the disease that, for many reasons, have not received the attention and resources that they deserve. So here, our mission is to highlight the potential of bioinformatics to contribute to the comprehension and the development of the treatments for this disease. So for the next 2 days, we will have the privilege to hear from experts, researchers, students, and who have dedicated their time and expertise to contribute with this complex mission. And I encourage each one of you to actively engage in these sessions, ask questions, and share your perspective. Because the strength of this conference lies in the diversity experience and knowledge that we can bring to the table. So thank you for being a part of this important journey, and I wish you all a productive and inspiring conference. First and foremost, it's an honor to introduce the professor. Uh, sorry, it's an honor to introduce the Professor Tetsu Sakamoto, a biologist and PhD in bioinformatics from the Federal University of Minas Gerais in Brazil. And through his career, he developed computational tools and database in the area of molecular phylogeny, such as taxon tree and taxonomy. He's a professional. He's a professor at Metrópole Digital Institute and a member of the Bioinformatics Postgraduate Program at the Federal University of Rio Grande do Norte. His main interests involve software and database development related to bioinformatics, the discovery of genetic factors associated with phenotype, evolution, machine learning, and structural biology. So today, Professor Sakamoto will share light on the latest breakthrough of bioinformatics and leptospirosis, uncovered the genetic basis of serology during his presentation. Professor, it's an honor to have you here, and the stage is yours.
Okay, okay. Thank you. Firstly, thank you, Matheus, for the nice introduction. And hello, everyone. Are you hearing me well? Okay. Okay. First of all, I'd like to say that it is a pleasure to be here to talk about my work. So, thank you for the organizing committee for the opportunity. And before I talk about the subject, the leptospira, I just want to add some more information to my introduction. So, as mentioned by Mateus, I'm talking to you from the city of Natal. And I think, as many of you know, Natal is a great city to spend your vacations. But some may not know, but Natal is also a city that offers several opportunities related to technological innovations. A large part of these opportunities is promoted by the Metrópole Digital Institute, which is the institute to which I am assigned and which belongs to the Federal University of Rio Grande do Norte. This large institute is the home of those seeking graduating in IT, in information technology, and welcomes more than 700 students every year at technical, undergraduate, and postgraduate levels. The Institute also offers an incubator structure for technological based companies and provides data center and high speed internet service for the companies located in its technological park. So it is within this innovative and technological environment that we find the Biome Group, which is a group of professors and students who carry out research in area of bioinformatics. The group is very interactive and form diverse backgrounds where together we carry out research covering the main subjects in bioinformatics. Myself specifically, I work mainly in the field of genomics, and one of my research interests is in unraveling the association between genotype and phenotype in a wide variety of organisms. I currently work with genomic data from bacteria such as Enterococcus and Leptospira to plants and animals like papaya, fish, and primates. As an example of a research question that we have in papaya, for example, I don't know if all of you are aware of it, but papaya has three sexes: male, female, and hermaphrodite. And the question here that we want to answer is, what is the genetic factors that determine the sex of a papaya? So to answer these and other questions, I use some statistical and evolutionary methods, as well as machine learning methods. So if you're passing through Natal during your vacations, I invite you to get in touch and get to know us, okay? So let's get to the business and talk about Leptospira. So, leptospirosis is a bacterial disease commonly known to be transmitted to human by the contact with the urine of infected rat. But actually, leptospirosis affects a broad spectrum of hosts. This includes humans and other animals, either domesticated animals like cows, pigs, dogs, etc., and also animals from the wild. It is also considered as the most extended zoonosis worldwide and represent a major public health challenge, mainly in those regions with heavy rainfall, floods, and poor socioeconomic conditions. The global annual incidence of leptospirosis is around 1 million human cases and about 60,000 deaths per year. Despite of that, leptospirosis remains a neglected and underdiagnosed disease. The causative agents of leptospirosis are pathogenic species of the genus Leptospira. Leptospira is a gram-negative bacterium with a very peculiar spiral shape. It is part of the spirochete phylum, which includes other species of bacteria with the same characteristic. Other clinically relevant species are in this phylum, besides Leptospira, are Treponema, which cause syphilis, and Borrelia, which cause Lyme disease. Okay, currently the genus Leptospira has 69 identified species, and phylogenetic analysis of these species from two main clades. One brings together the pathogenic species that are in this part of the tree, and the other in the other clades here brings together the saprophytic species, which are in this part of the tree and are species that do not cause disease and are found in the environment. Okay, the main symptoms of patients with leptospirosis are high fever, vomiting, and diarrhea. And if not treated properly, it can develop into a severe conditions such as kidney or liver failure or meningitis. Uh, Treating leptospirosis is not a difficult task. As a bacterial disease, it is controlled using antibiotics. But the problem here is that the symptoms of leptospirosis is very similar to other febrile viral disease, such as typhoid fever, acute sinusitis, influenza, and dengue. So until the discovery and the appropriate treatment of leptospirosis, the patient will continue having these unpleasant symptoms, and if not treated, it could evolve to the severe conditions. In commercial animals such as cows, sheep, and pigs. A major problem with leptospirosis is its late detection. In these cases, owners end up detecting the infection after observing the symptoms which cause major losses, such as low productivity and a high rate of abortions and stillbirths. Okay, in this sense, the prevention through vaccines is the most appropriate way to control the disease. Vaccines can prevent severe conditions of the disease and economic losses for producers. However, although there has been a great effort to develop vaccines against leptospirosis, there is still no effective vaccine for its control. The vaccines that exist of leptospirosis are only for animals and are based on inactivated bacteria, the bacterine. Although they confer immunity on the animal, this immunity is short-term and is also specific to certain strains of leptospira. So one of the challenges with vaccines is that we need one vaccine that provides protection against a broad spectrum of leptospira strains. But this is a very challenging task, and I want to show you why it is so challenging. So, well, the reason why it is so challenging to develop a broad spectrum vaccine for leptospirosis is related to the lipopolysaccharide or LPS of this bacteria. LPS is a structure made up of sugar in one part and lipid in other part that is anchored to the outer membrane of gram-negative bacteria. LPS is the main antigen that activates the body immunity, and this happens with other bacteria too. But peculiarity of Leptospira LPS is that it is large, complex, and very highly diverse compared to other bacteria. The LPS synthesized by each Leptospira sample can differ in size and composition. As a result, an antibody that has been produced to bind to the particular LPS will not bind to others. So Leptospira samples are also usually classified according to the serology, which has to do with the structure of the composition of the LPS, in serovars. And to give you an idea of the serological diversity of Leptospira, there are currently more than 300 identified serovars. These serovars can be grouped into serogroups according to their antigenic similarity, and there are more than 30 serogroups described so far. Okay, and in other words, we can find, for example, we can find, for example, two samples here from the same species, Leptospira interrogans and Leptospira interrogans here, that are part of different serogroup, and another bacterium from a different species, for example, Leptospira borg-peterseni here, that is part of the same serogroup of this sample here. So taxonomically speaking, bacteria from the same species, these two one here, are more closely related, but in serology, samples from the same serogroups here are closer, even though they are from different species. Well, this is one of the big reasons why it is difficult to develop a broad spectrum vaccine for Leptospira. What is usually done then is to carry out epidemiological studies in the regions of interest and assess which servers and serogroups are most prevalent and develop vaccines for these samples. At this point, we are faced with yet another challenge in the world of leptospirosis, which is to identify the serology of the samples. The serological classification of the sample is carried out by a method called microscopic agglutination test, known as MET, which is a immunogenic test that works as follows. You collect here serum from a patient or animal infected with the leptospira. In this serum, it is expected to have antibodies produced by the host in response to the strain of leptospira that infected the host. This serum is then applied to leptospira samples that have been isolated, cultured in the laboratory, and that have been serologically identified. We then observe if there is an agglutination effect, agglutination reaction, which indicates here that the antibody produced by the host is binding to the leptospira sample. So if agglutination occurs, it indicates that they belong to the same serological group. So the leptospira samples strain that is infecting this patient could be this one here from the serogroup Serroi, Serovar, Hardisho, for example. Well, the MET method is interesting, but it has several disadvantages. Firstly, it requires an adequate infrastructure to maintain the leptospira cultures, and this is not a reality for many underdeveloped countries, which are the most affected by the disease. Secondly, the methodology requires a few days or weeks to get the result. Third, a specialist is needed to interpret the results. And lastly, the methodology may not be as accurate because of cross reactions, where agglutination reactions can be observed not only in one but in several leptospira samples from the library, which makes identification a little more difficult. So there are two major challenges in the world of leptospirosis: the development of a broad spectrum vaccine and the development of faster and easier methods for serological identification. One thing that could help solve these challenges is to learn more about the genetics behind the serology of these bacteria. So, since knowledge on this aspect is still limited in Leptospira, I set out on a journey together with some students and collaborators to try to unravel the genetic patterns behind the serological classification in Leptospira and try to propose new methods for serotyping these bacteria. So, we took an initial step here to unravel the genetic basis of Leptospira serology by initially taking a serogroup, the Serroi serogroup. This serogroup is well known among those who study Leptospira, since a sample of this serogroup was used in the past to unravel some of the genetic factors that affect the serology. This serogroup mainly infects cattle, and it is interesting because there are 42 samples of this serogroup with genomic data, and it covers 3 different species. In this first step, we then asked whether there are any genes that are specific to Serroi serogroup samples. And to do this, we carried out some analysis using blast identity calculation and the Patrick database. Basically, what we did at first was to choose a reference sample from this cell group Serhui. In this case, we chose the strain Norma, which belongs to the Leptospira interrogans species, and do a comparative genomic analysis with other samples that are from the cell group Serhui here, and with other that are not from the cell group Serhui. In preliminary analysis, comparing this reference sample with these other 6 samples listed here, we obtained the following figure, this one here. So the figure here is a circus format where each strand represents a genome, okay? The 3 innermost strands here are the samples from the other cell groups, and the following 3 strands here represent the samples from the same cell group, from the cell group Serhui. And the reference sample is represented by this strand here, the outermost strand, okay? And the different colors here correspond to different similarity values, where the bluish color represents high similarity. So we could observe here that the samples that are from the same species as the reference, here and here, okay, are those samples that show it the highest similarities as expected. And the idea here is to find a region where the Serhui cell group samples share a greater similarity between them than between samples from the other cell groups. Note that in this sense, there is a small region right here, where we can verify the existence of genes in the reference sample that show a higher similarity only between samples from the cell group Serhui, but not from the other samples from other cell groups. Note that even the samples from the same species here as the reference, they do not, they present a low similarity for those genes. These are certainly good candidates for defining the Serhui cell group. And in fact, when we analyzed this region, we found that it encompasses a locus called RFB, which is made up exactly of genes that encode enzymes responsible for LPS biosynthesis. The figure here shows the RFB locus of the reference sample. In this sample, this locus is made up of 75 genes, and most of which encode proteins that add or modify sugar, such as transferase, oxidoreductase, transaminase, isomerase, and others. The genes here in the gray box are those highlighted in the previous figures, which show higher similarity between samples of the same cell group than in others. With this first evidence that there are genes of the RFB locus specific to the Serhui cell group, we decided to carry out a broader analysis, performing a similarity analysis of the genes of the RFB locus of the sample representing the Serhui cell group with the more than 700 leptospira genomes available in the NCBI. The result was this heat map. And in this heat map, each row represents a sample and each column represents a genes. And the genes delimited by this ruler here are those genes that belong to the RFP locus. The color of the heat map here represent the similarity between the proteins. The more bluish tones indicate high similarity, while reddish tones indicate low similarity. It can be seen at a glance that most of the samples here, analyzed here, do not have the same gene composition as the RFP locus of the reference sample, and that all of them are not from the Serhui group. The few samples that do have similar composition are higher up here in the figure. So zooming in on this part of the figure, we can see that all the Serhui group samples have a similar gene composition to the reference sample, but in addition to the Serhui group, we also found that the samples from the mini and Hebdomades group also had a very similar gene composition to the group Serhui. This led to some doubts in the analysis, but upon further study on the literature and the history of the serological classification, we found that these three serogroups here were actually part of the same serogroup, and that it was broken up in 1982, which made perfect sense for us. So from then on, we were convinced that the gene composition of the RFP locus was closely associated with the antigenic similarity of this bacteria. After this, we did some further analysis to see if it's still possible to distinguish these three serogroups, Serhui, Mini, and Hebdomades, through the gene composition of the RFP locus, and we found that it is possible. In the RFP locus, there is a small region between the ORF13 and the ORF18, whose the gene composition here varies according to the serological classification. This region is schematized here in this figure, where we can see the gene composition of this region in some samples from the serogroup Serhui, Mini, and Hebdomades. The other figure here shows a similarity analysis similar to the previous analysis, but focusing only on this region and using sample from Serhui, Mini, and Hebdomades serogroup as a query. You can see here that in this region, samples that are part of the same serogroup, here for example from the serogroup Serhui, have high similarity, here from the serogroup Mini and here from serogroup Hebdomades, indicating that this region is decisive for classifying the sample in one of the three serogroups. There are some few exceptions like this and this here, but we later found that these samples cross-reacted, and this may have led to a misannotation on these samples. For this region, we called it as SVR, which stands for SHA, Serhui, Hebdomades, and Mini RFB variable region. So knowing all of this, an interesting thing that we performed here was also to attempt to classify those samples that do not have a serological classification. Among the samples that we used in this study, more than 300 samples do not have the serological classification. So we checked which of these samples had gene composition similar to that of the Serhui, Hebdomades, and mini serogroups, and tried to serologically classify them using only the information from the gene profile of the RFP locus. Of these 340 samples, 51 samples have similar gene composition on the RFP locus, and we could classify them into one of these three serogroups only based on this small region, on these SVR regions of these samples in the RFP locus. So the figure again is a heat map, very similar to those heat map that I showed previously, focusing only on the SVR region. So we can note here that we could classify each of the sample here in one of the serogroups, and we can note here that we do not found any overlap classification for those samples. This all lead us to conclude that these genes are promising candidates for use as genetic markers for the serological classification. The whole story of this work was published in this article last year, so everyone interested can look for more details in it. Well, from this first study, we were able to identify some important molecular markers for identifying the Serhui, Mini, and Hebdomades serogroups. But what about the other 27 serogroups? Can I establish any antigenic similarities between them and find, for example, the regions that characterize each serogroup? So to try to answer this question, we set out a Mayer analysis that would allow us to verify, to observe the genetic and structural diversity of the RFP locus in leptospiros in general. So to do this, we first carried out an orthologous cluster analysis involving more than 700 leptospira samples using OrthoFinder. Then we identified those orthologous groups that consists of genes that are part of the RFP locus, and finally we carried out a hierarchical cluster analysis to verify the similarity of the gene composition between the RFP locus of the samples. So the result was this large heat map on the right. And this heat map, again, the row represent the samples and the columns represent the orthogroups. The dark part of the heat map represent the absence of the gene in an orthogroup in a sample, and the lighter part represents the presence of the genes in a sample in an orthogroup. In this heat map, in this analysis, we can see that the samples can be grouped into six defined clusters, okay, where samples in the same cluster theoretically share a high degree of similarity of the gene composition of the RFB locus. Note this cluster here. It's a little small, but this cluster are putting together the samples from the Serhui, Mini, and Hebdomades serogroups. We can see also that most of the samples from the same serogroup are grouped together in the same cluster. For example, the for example the icterohemorrhage cell group, parogenes, and canicola. The samples belonging to these cell groups are all clustered together in this cluster here. And another interesting observation is that these first 5 clusters here are all made up of pathogenic samples from the P1 clade. The last cluster here is mainly made up of saprophytic samples, but we did find some few pathogenic samples too. We were also able to group the orthogroups into 13 clusters. Okay, some of these clusters are specific to one of the samples clusters, like this one, this one, and this one. And there may be interesting genes that characterize each of the clusters. We have also schematized the RFP locus between samples from different cell groups, as shown in this figure. And we have also verified that samples from different cell groups, but which are part of the same clusters, all of them share a similar gene composition between them. This also indicates, as with the Serhui, Hebdomades, and mini cell groups, that samples from the same cluster can share an antigenic similarity between them. One additional thing that we made here is that these 2 first clusters here, we decided to put them together because the gene composition of samples from these 2 clusters are very similar. So we labeled each of the clusters as classes, and in the end, we suggest that Leptospira samples can be classified into 5 classes according to the gene composition of the RFB locus. Okay. We also carried out some functional analysis of the proteins encoded by the RFB locus. To do this, we extracted the amino acid sequence of proteins encoded by the RFB locus from the representative of each cell group and performed some functional annotation using the superfamily database. In this analysis, we were fortunately able to annotate most of the proteins, approximately 82% of them, and we verified the existence of at least 69 functional groups, and some functional groups are found in all of the samples, but most of them proved to be specific to a sample or to a group of samples. There were also some functional groups that are found in great abundance. Of the about 2,000 proteins that we submitted for the annotation, more than 300 proteins were annotated as nucleotide diphosphosugar for example. These most abundant functional groups are generally proteins that catalyze sugar modification and addition, again demonstrating the complexity of the structure of the Leptospira LPS. We also observed that some of these abundant functional groups are differentially distributed between samples of different classes. And this again reflects the structural variability of the LPS that samples of each class can synthesize. A more comprehensive analysis to verify the distribution of the functional groups among the samples were made using the hierarchical clustering. Here we could observe functional groups that are found in most of the samples. Here are those uh, functional groups that are specific or found in a few samples here, and we have those functional groups that are specific to a class or shared with some classes. This also is demonstrating again the richness of the functional groups found in the RFP locus and the conservativeness of some functional group among samples belonging to the same clusters. Okay, so this story is also was published initially in form of a preprint, but it is currently being reviewed, and we hope that it will soon be published in a good journal. Finally, after studying the RFP locus extensively, there is nothing more appropriate than proposing some molecular markers to, for example, amplify them with PCR and try to identify the serogroup of a sample based solely on molecular methods. The choice of these markers could be made by manually analyzing the similarity results between the genes, as was done in the first paper, or even by analyzing the orthogroup table. But as you have seen, the Leptospira RFB locus is very diverse and some are quite extensive. So performing this task manually is very laborious. So here we use some machine learning techniques to automate this task of selecting the interesting markers for serological classification. So as the genetic patterns that characterize the serogroup are not so complex, we used here some simple machine learning methods that allow us to interpret which attributes it considers important for classification. We then use it here the decision tree method, which is a very simple method that set up a tree structure like this here, where each node of the tree represents a condition based on an attribute, and that takes you down to different paths until you reach a classification of this sample. So to apply this method to our problem, we use it as input a table similar to a similarity matrix between genes, like the one presented throughout the paper, where each sample is labeled for a class or serogroups. The strategy here was to apply this method in two stages. The first was to generate a model to classify a sample into one of the 5 clusters of pathogenic Leptospira. So the labels here were the classes. And then to generate a model for each of the classes so that it classifies the sample into one of the serogroups. So this is a work in progress, but we have some very promising preliminary results. where here I demonstrate a decision tree classifying a sample into one of the five initial clusters. So in this tree, each node represents the decision of the existence of a particular gene. So in this part of the tree, for example, we have the separation of my samples into two groups here based on the existence or not of this gene here. And in each node here, we will have this evaluation until we reach the end of the tree, where the method will return you a classification. This tree, so this tree classifies the samples into one of the five clusters. And here we have another tree which classifies a sample from the class 1 into one of the 8 groups that is present in these classes. Again, this work is in progress, but it greatly facilitates this step of selecting these markers. And the idea is to make the finding of these studies applicable in the clinical labs. So finally, to conclude my presentation, so the extensive analysis of the genomic data allowed us to do several things. One was to verify and confirm that the gene composition of the RFB locus is strongly associated with antigenic similarity in the genus. We were also able to contemplate the pattern of conservation and specificity of the gene composition of the RFB locus in Leptospira. And also we could suggest, and also we could suggest here a broader classification of serology between samples. And also we were also able to verify that it is possible to use molecular methods to carry out the serological classification. And the better understanding of the genetic basis of leptospira serology promoted by these studies could lead also to an improvement in leptospira serotyping by using molecular methods, and also could help in the development of a broad spectrum vaccine. Now, with the notion of the genetic basis of the leptospira serology, it also opens up to new research horizons. In the wet lab, for example, we can propose some knockout experiments of these genes, for example, to verify the essentiality of a gene in manifesting the phenotype of a cell group. In bioinformatics, we are proposing here some studies to predict the three-dimensional structure of the genes that make up the RFB locus, and also try to infer the LPS biosynthesis pathways. This would be interesting for predicting, for example, the structure of the LPS. It is also of great interest to carry out some evolutionary studies of the genes that make up the RFB locus in order to understand the dynamics of how a new set of groups or a new servers arise in this species. So here are my thanks to the entire lab group and my collaborators. Thanks to the Biome Group, IMD, and the UFRN and also to the agents who promote research and the student grants. And thanks to the entire audience, my contact is here, and I'm also open to collaborate. So if you have some research question about genotype and phenotype association, please feel free to contact me. So thank you.
So thank you, Professor Sakamoto, for the amazing presentation. And now let's
Thank
open
you.
for the floor to the audience. So if you have any question, please share them in the chat on YouTube, and we will navigate through them during the Q&A session. We have 10 minutes for this. So we already have some questions here. I will project and read them for you. Uh,
Okay.
the first
Okay.
one is from João Roberto. He says, hello, Professor Tetsu. Could you explain a little bit more about the techniques and tools used for genomic visualization?
Okay. Okay, João. I don't know which visualization are you referring, but if you're referring the, let me uh, let me see here. If you're talking about uh, this circus format visualization, this visualization was made using the Patrick database. For it's a visualization that the Patrick database automatically generates for you. If you're talking about the scheme of of the locus, for example, here and here, it was made using a Perl script that one of the paper, uh, one of how can I say it's a Perl script that one author of a paper made available for us. It not a published work, but it's a script that worked very good for us. I don't know that if I answered your question.
Yeah, I think you got a perspective of how to build these figures.
Okay,
Thank you.
okay.
So the next question is from Gabriel Matsuguma. He actually anticipates your final slides and he asks, are there current trends in machine learning for the scalability of these biomarkers analysis to big data analysis?
Oh, okay. So it's a very good question, Gabriel. Thank you for the question. And yes, I think that the actually the machine learning techniques help us to interpret our results when we are dealing with a large scale analysis, as I showed here. If not, if it was not the machine learning, I would have to do this analysis manually. So machine learning help us in this sense. If you're talking about an analysis uh, that is it's like a big data scales. So yes, we have some trends about it. We could use some how can I say some techniques that allow you to do some parallel analysis, like some the library from the Spark. I don't know if you know Spark, but is a method that can parallelize some task that we use in machine learning. So yes, there is some trends, and we expect that we have some problems that we cannot use the traditional machine learning techniques to analyze, to interpret and analyze our data. I
Okay,
think
thank
it is.
you. And we have one more question. Raissa Medina asks you, in your opinion, what was the biggest challenge on this project?
Okay, Haisa, it's a very good question, and I think that the biggest challenge here is the data. So, as you saw here, for example, we analyzed the 700, more than 700 genome samples from Leptospira, but as you see, most of the data, the data that we have here, do not have, for example, the serological classification. So having a good quality of this genomic data, and also the metadata about the serological classification is our biggest. I don't know if it's is the biggest, but is one of big challenge. Another challenge that we are facing now is to confirm in the wet lab and our findings. So is another challenge to have some financial support from the agencies to have material, to have infrastructure to do this kind of experiment. I think it's a very promising finding here, but it is quite difficult to have some financial support in this sense. So I think that this is our biggest challenge.
Okay, thank you. And actually, I have one personal question.
Okay, okay.
Sometimes we have some resistance from the experimental researchers to work with bioinformatic tools. So, what is the current status of the collaboration between wet lab and dry lab in the scenario, in your scenario?
Okay, okay. Uh, I think that in our scenario, the the collaboration between the wet and the dry lab uh, is um, is starting to be uh, to be aligned, because uh, I don't know, but the analysis that we performed here is relatively simple analysis. It's some blast analysis with some comparative genomic analysis. But as you saw, these findings and other is very recent. So I think that in the past there was no conversation between those two areas, the bioinformatics and the wet labs. But now it is starting. They are starting to meet each other to try to solve this big challenge. So I think that from now on, this kind of collaboration will be more intensive. So I hope that we could uh, we could have more innovative solution for to control the disease.
Yeah, it is the future, right? Um,
Yes.
So I think that is the end of our questions. So I want to thank you once again for your time and for the amazing speaking. So now we go to our break and we'll get back at 3 p.m. Right? Goodbye, everyone.
Okay, thank you, everyone. Thank you. Thank you, Matheus.
Yes. Thank you. Bye bye.
Bye.